All right, so good morning, everyone. So today we have the pleasure of our guest speaker, who is Dr. Lily Guenther. And so I want to give you a little bit of an introduction as to her accomplishments. She obtained her bachelor's in history at Brown University, then went on to obtain an MD at SUNY Downstate Medical Center College of Medicine, then went on to do a pediatric residency at Boston Combined Residency Program, Harvard Medical School, then a pediatric hematology oncology fellowship at Dana-Farber Cancer Institute, Boston Children's Hospital. After that, she was an assistant member, excuse me, an instructor, or part of that instructor Department of Pediatrics, then an attending pediatric hematology oncology, now an assistant member of Department of Oncology, Division of Molecular Oncology, St. Jude Children's Research Hospital. So a little bit of a context about the research that she does. So her laboratory research focuses on the investigation of novel molecular targets related to replication stress and DNA damage, with particular disease interest in Ewing sarcoma and osteosarcoma. Her goal is through mechanistic investigation and validation with goal of moving forward genetic dependencies for therapeutic interventions. She applies genome scale CRISPR-Cas9 screening and other modalities to answer key biologic questions in sarcoma models, including targeted drug synergy and resistant investigations. She has been very active in the academic community, served as a grant reviewer, journal reviewer, and has engaged in a number of teaching activities. Also, participates in the supervision of pediatric residents and pediatric hematology oncology fellows, in addition to serving as a mentor of research students. She's obtained a number of grant awards from private and federal funding agencies and has had over 30 peer-reviewed publications and lecture presentations. So with that introduction, I turn it over to our guest lecturer.
Thanks, Dr. Tanoka. That was a really gracious introduction. And it's so nice to be with you all today. And I understand that you've gotten a really nice breadth of guest lectures in this course, along with a lot of biological insights from journal reviews, et cetera. And so I hope that I can add guys are studying. This is a small group, so I don't mind. Do you mind if I take, like, if there are questions that come up along the way, do you mind if I
Yeah, that's good.
get, if
Along the
people
way.
want to interrupt me, I'm fine with that. So please feel free to speak up, or my guess is that Dr. Tinoka will monitor the chat for me a little bit. And then, of course, I'll take questions at the end if that's preferable. So let me try I just practiced this with Diane, so hopefully, can you see my PowerPoint?
Yes.
Okay, perfect. So the title of my talk today is going to be From Functional Genomics to Novel Therapeutic Targets in Pediatric Sarcomas. And I first wanted to tell you just a little bit about me, not about my, that what Dr. Tanoka covered in the background. First, I am not a chemical biologist, and I know that you are all chemistry students, but I have really had lots of wonderful intersections with chemical biologists along the way. And so I'm going to mention those throughout my talk today. I really began my path to medicine as a clinician. And as you heard, I only have an MD. I'm not an MD PhD. And so I sort of fell in love with science along the way through my training. And that's what I've set out to devote my life to. But it really was a sort of a curvy a journey. And I'm happy to talk to any of you who are sort of thinking about what you want to do after school. I am a physician scientist, which means that I also care for patients, and that consists of taking care of patients with solid tumors and brain tumors at St. Jude who are admitted to the hospital. That can be anything from chemotherapy to new diagnoses that are acute enough that require admission for workup. And one of the diseases that I care for a lot, that I'm also very interested in the lab, is osteosarcoma. I'm showing you a picture here of a patient with osteosarcoma who's had a very interesting limb salvage procedure. She had a tumor in her right leg that actually was in her femur, so in her upper leg, shown here. And she had something called a rotationoplasty, which is an interesting procedure because it basically uses physics to determine the best way to preserve function in this case, this child's life, where they actually took her lower leg and reattached her tibia and fibula up here, sort of around where this brace is. And if you can imagine it, her leg is turned backwards. So her ankle and the base of her foot actually form the part where her knee is hitting that prosthesis. So that's actually a lot more of a stable way for kids to have function. And, just to provide you with a glimpse of hope, we have a lot of kids who have surgeries like this and they go on to run professionally, play all kinds of sports, and are incredibly active. So I really love what I do clinically. Let's see, can I go forward? So the goals of my lecture today are first to give you a background on my lab and the diseases I study. I want to give you an overview of functional genomics and CRISPR screening, on some background on a project that integrates those technologies, which is called the Pediatric Cancer Dependencies Accelerator, or PEDDEP, which I'm very involved in, and really place that work into the context of my lab and what I do. And then I'm going to tell you a couple of scientific vignettes about how we leverage this data in our own lab. So the first disease I'm going to tell you about today is a bone sarcoma called Ewing sarcoma. It's a rare disease with a very small incidence. As you can see here, it's the second most common malignant bone tumor that's seen in children and young adults, and has a peak incidence in adolescence around the age of 15. Boys are more affected than girls in slight predominance, and it mainly affects children of young adults of European descent. One of the interesting things about this disease is that it really does not affect any individual of African descent, which we still don't understand why. We think that maybe there are SNPs in the genes that have been inherited in that population over time that are protective, but nobody has really been able to understand why. It's about 75% localized disease at diagnosis, which we do fairly well with. We cure about 80% of those patients, but in the 25% that are metastatic or those who relapse, they actually fare much more poorly. It's treated with basically everything we have in oncology: dose compressed, multi-drug chemotherapy. We do surgeries that are quite aggressive in these patients, and we treat them with radiotherapy for local control. And as you can imagine, there are a lot of long-term toxicities from all those therapies from the children that we cure. And there are really no molecularly targeted therapies available for this disease. And I'm showing you sort of a classic X-ray here of a patient who was diagnosed with Ewing sarcoma, which shows you can see this sort of line of lucency here. This is a classic finding that's called onion skinning. It's when the periosteum of the bone is sort of lifted off the rest of the bone by this really aggressive tumor. And you can see all of this like soft tissue involvement. You can see it even better here, sort of arising from the bone, but extending into the soft tissues. These children metastasize most often to the lung. And so I'm showing you a chest CT here. And then this is also an MRI, which also is used to stage these patients and plan for surgery. And you can see, again, the sort of soft tissue infiltration and edema that's associated with this bony disease that's right in here. This is just a bone scan that's showing you again how hot these tumors are that light up, in this case, in the lower leg, so in the tibia in this patient. And when you look at these cells under the microscope, you see these sheets of what are called small round blue cells. Basically, they look very uniform. At higher magnitude, you can see the uniformity of the cells and the size of the nuclei. They stain very positively for CD99 on their cell surface, which is one of the markers that's used to diagnose these patients. And then they have a very classical fusion, which I'm going to tell you about in the next slide, that's diagnosed by FISH, or fluorescence in situ hybridization, which is a technique that uses fluorescent antibodies to detect a protein. So in this case, there's a fusion which is dividing the EWSR1 gene. And so you can see a normal cell here where these two probes at each end of the protein are together, which is how you would expect it to be. But here, the two ends of the protein are divided. And so that's what's seen in malignant cells, and that's what's used very often to diagnose these patients. So we know a fair amount of the biology about this disease, but we don't know what the cell of origin is of these cancers, which is a really interesting thing. There's a hot debate. Do they derive from neural crest versus a mesenchymal lineage? And there are pros and cons to each of those theories that I'm not going to really get into, but it is one of the things that is challenging about this disease because, for example, since we don't know the cell of origin, we don't have really good mouse models of this disease. As I told you, it's driven by a pathognomonic fusion transcription factor. And so this is most commonly called EWSR1 Fly1. And I've included this chart here of the different fusion partners that are seen. Usually it's between this FET RNA binding protein, typically, as I said, EWSR1, but can rarely be consistent of other FET protein members. And usually the partner is an ETS transcription factor, or always it is an ETS transcription factor. And the most typical here is Fly1, but you can see that there are others that do appear in this disease. And what this does is it activates the ETS transcriptional, the transcriptional active domain of the ETS transcription factor. And so it really drives abnormal gene expression in these cells. And there, as I said, vast transcriptional dysregulation in these cancer cells. And the way that this happens is because the transcription factor, as you've probably learned a little bit in the biology in this course, the transcription factors are in charge of helping to drive changes in chromatin that lead to activation or repression of different genes. So you can see that when EWSR1, Fly1 is expressed, so the fusion oncoprotein in these cells, this causes some genes that are not typically active. They're typically closed chromatin that's not transcribed and not activated, gets aberrantly activated, and some that are active become repressed. So it can be both activating and repressing of gene expression. There are very few recurrent somatic mutations in this disease, and I've just mentioned two here, including one that you've probably all heard of, P53, but that's relatively rare. So then I'm going to talk to you about a second disease, and I think it's important to draw the distinctions between these two because they are quite different. This is the most common malignant bone tumor in children, and it's called osteosarcoma. Also peaks in adolescence. And this disease is actually associated with quite a few predisposition syndromes, and we're actually learning more and more about predisposition associated with this disease. The most common one is one that you've probably all heard of, which is called Li-Fraumeni syndrome, or LFS, that's associated with germline loss of TP53. So those patients get very aggressive osteosarcomas and very often die from metastatic osteosarcoma. Metastatic disease is the most important indicator of outcome, and really we do not cure patients with metastatic osteosarcoma. It is a very rare thing when we do, and so I just want to emphasize how much these children are in need of new therapies. They also get very aggressive chemotherapy and surgical resections, but they're considered radiotherapy resistant, and that's probably because of the calcified matrix that these tumors lay down. And so I'm showing you an H&E stain here of osteosarcoma cells. And you can see all of this sort of pink stuff in the middle of the cells. And that's all abnormal calcification, like abnormal bone that's being laid down by these cells. And that actually is a key pathologic finding. If you see that, that is a diagnosis of osteosarcoma. Furthermore, the treatment for these children has not changed since the 1980s. So really, chemotherapy is the same as it's been for many years, which is different from other diseases. Also metastasized to the lung, also have lots of soft tissue involvement. And this is also like another sort of classical finding, that this sort of cloudy appearance that you can see all the calcification surrounding the bone. So a little bit different from the onion skinning I showed you with the Ewing sarcoma. In terms of the biology, these tumors are quite different from Ewing sarcoma. I told you that those tumors have hardly any molecular findings. These are a very chaotic genomic disease with complex chromosomal rearrangements. So they're characterized by things like chromothripsis, which are gene shattering events, which lead to vast copy number and structural changes in the DNA of these cells, different gene disruptions, copy number changes on their own without chromothripsis is also seen, and gene deletions, as well as contagious, which is also another type of gene chromosomal shattering event. There's a lot of heterogeneity because of all these different events from tumor to tumor, as well as intratumoral. And we see classically more mutations than we do in osteosarcoma. So P53 and RB mutations are very common, as well as MYC, which is a well-known, well-studied oncogene, which you've probably heard of, is commonly amplified in this disease, as well as CDK4. Um, and I just want to show you a karyotype of a normal cell and contrast that for you just to get a visual of how chaotic and messed up these cells are. So they're really, they have really abnormal karyotypes. You can see just how many copies of the different chromosomes these cells have. And so it's really a miracle that these cells are able to divide, quite honestly, I'm sort of in awe of it, but it's pretty nasty. Let's just leave it at that. So, as I've sort of emphasized, we have some room for improvement in bone tumors. This is SEER database data from 2019, which surveys across the United States, and looking just at pediatric diseases, 5-year overall survival rate. And you can see in leukemias that we do, and lymphomas, that we do fairly well in those tumor types. And then in the solid tumor space, which is the second half of this chart, bone and joint cancers, which are primarily the two diseases that I just described to you, fare the poorest out of all of the different solid and brain tumors that are listed here, the categories of tumors. So we really need to do better. And as I've mentioned, they're the poor actors of these metastatic patients. So I'm showing you two Kaplan-Meier curves here, which is a well-defined way to show long-term survival from clinical trials. And the treatments here don't really matter. What I'm trying to emphasize is that the orange and the yellow are the metastatic disease patients, and they do very poorly. And the same is true in osteosarcoma. They do very poorly. So as I'm going to introduce to you functional genomics and CRISPR screening, I want to emphasize to you one sort of principle of pediatric cancer genomes, that in general, pediatric cancers are quiet. And what do I mean by that? I mean that their somatic mutation burden, which is what's shown here on the y-axis in this graph that's from over 12 years ago, we recognized this systematically, that pediatric tumors, which I've circled for you here in red, tend to have the lowest mutational burden compared to all of these other tumor types that are adult tumor types. So really, they can be very nicely distinguished from adult tumor types in that way. They have fewer mutations in their cells. This is a more recent paper that looked into more detail at pediatric tumor types. And so all of the purple sort of columns here represent pediatric tumor types. The green are all adult tumor types here. And again, this is looking at mutations per megabase. And the reason why I wanted to show this to you is because you start to know that you start to realize, and we've started to realize recently, that there are some exceptions to this rule. And osteosarcoma in the red box is one of those exceptions, which probably doesn't surprise you because of the genomics I just showed you that are so scrambled. But but osteosarcomas tend to be towards the adult tumor types in many cases, not all. But sort of the other ones that are here are like high-grade gliomas in brain, which we tend to lump together a lot with osteosarcomas actually because they both have lots of mutations. Whereas Ewing sarcomas tend to be more quiet, like the typical pediatric tumor type. This I'm showing to you here is actually cell line data because I'm going to show you all cell line data from my lab. So these are models that are used to study cancers. And this is sort of a mishmash of pediatric and adult. And this is looking at copy number aberrations or CNAs that are seen in these tumor types. And again, I'm highlighting for you in blue here, Ewing sarcoma and then osteosarcoma. And you can see again that osteo is way higher than Ewing. And so that really, there's a spectrum. And I should say all of the pediatric are colored on this graph. So you can see there's sort of a spectrum here, but osteosarcoma tends to be towards the top. So there are sort of, challenges, as I've alluded to, in developing targeted therapies for children. First of all, I want to sort of celebrate the successes, which have been the BCR-ABL inhibitors, which you've probably all heard of, that are used in adult CML, but also in pediatric CML, which is a disease we do see, and some ALL children with ALL, acute lymphoblastic leukemia, also have what we call BCR-ABL-like disease, and we also use those inhibitors very successfully to treat those children in a targeted fashion. Another classical example of a major success in pediatric solid tumors has been the introduction of an NTRK inhibitor. And NTRK is neurotrophic receptor tyrosine kinase. And these kinases tend to be rearranged, sort of similar to the EWS fly rearrangement that I'm showing here. There's a gene rearrangement that involves NTRK, and that can be targeted with drugs. And those tumors go away completely. And now we're able to observe some of these children who are some of our youngest kids who develop solid tumors off therapy for a long time and they do not recur. But these kinase type of alterations are very rare. And this 25 to 30% is not only kinases, but that is the max that we see targeted therapy recommendations for in pediatrics. And really the recurrent alterations that we see in the majority, in the other like 75% of patients, are either in lineage transcription factors, which as you may know are very difficult to drug, as well as in these fusion oncoproteins, which like EWS fly that I showed you earlier is also very difficult to drug. These are also rare diseases with few samples available. And so one sort of pathway that has been taken that I've been very engaged in my lab and since I've been a postdoc is using functional genomics to try to understand new vulnerabilities in these types of cancers. So I'm showing you a schematic for a project that's called the DepMap. The pediatric arm of the DepMap is called the Pediatric Dependencies Accelerator, or PEDDEP, that involves the Broad Institute and Dana-Farber in Boston, as well as St. Jude. And this has profiled, this database has profiled over 1,400 cancer models to date, including about 300 pediatric models. So a pretty robust assortment of pediatric models, including now we have 45 osteosarcomas and about 60 Ewing sarcomas, which is really nice for very rare diseases. And what we have done with these cell lines is systematically performed CRISPR screens, and I'm going to get into CRISPR screens in more detail in the next slide, as well as doing other molecular characterizations of these cell lines, including RNA sequencing, whole genome or whole exome sequencing, copy number, and DNA methylation profiling. In some cases, epigenetic marks have been profiled by CHIP or cut and run. So there's just a lot of molecular data that's available for these cell line models. And in addition, we've done more and more drug sensitivity screening over the course of the time this project has been active. And so this is mostly looking at repurposing established drugs, but also some compounds that don't have as many indications, or the mechanism is unknown. They've been profiled here. We're also starting to do combination drug screening, which is really exciting for some of the targeted drugs. So, and this is my slide to just remind you that this is not only being done at Harvard in the DepMap, but this is also, these types of analyses are being done across the world. The Wellcome Sanger Institute is a big institute in the UK that has done a ton of functional genomic screening in cancer models. And there's a big consortium in Victoria, Australia, that is also doing the same thing. And they've all put out big landscape papers of their findings that are interesting to read if you are interested in this sort of thing. So I think probably all of you know what CRISPR is, but I'm going to quickly go over it. So it's I think a lot of people call it a molecular scissors, and it's basically harnessing a technology that, well, it's a very ancient way that bacterial cells overcome viral DNA insertions into the bacterial DNA. Whereby cells are, where we use it as a tool in human cells, whereby we infect cells with this enzyme that was traditionally used in bacteria called Cas9, which is used it basically gets recruited to these sites on DNA called PAM sites, which are shown here in orange. And we infect cells using lentivirus with a guide RNA that basically includes a PAM sequence, as well as a series of nucleotides that are specific for one gene in the genome. And you can use this to actually recruit Cas9 to that site and cause cutting. And typically, the classical way this has been used is to delete a gene's function, and that's primarily what I'm going to talk about today. But it's also being used to do things like insert genetic code for other genetic diseases besides cancers, for example. So when we talk about large-scale CRISPR screening, genome-scale CRISPR screening in something like the DepMap, what we're talking about is using a pooled library of CRISPR guides, so those guides that recruit Cas9 to specific sites of DNA that represent all the genes across the genome. So basically about 20,000 genes across the genome are targeted, and this is all in a pooled fashion. So all of these individual CRISPR guides have a barcode on them, a genomic barcode that allows you to differentiate one from the next. So it's really very cool, because then you can allow cells to grow with this library infected, and then you can do sequencing for the barcodes to identify what's left. This is typically done at 21 to 28 days. So you let the cells grow with this CRISPR library infected into them. And then you can see one of three things. You can see either the cells, replicate as you would expect them to, similar to your input. So they're identical in case here in the orange. You can see that maybe those barcodes are overrepresented compared to input. So that would suggest a growth advantage of knocking out that gene's function. Or you can see dropout, or what's called or less expression of that barcode, which is shown here in blue, which are really what we're focused on, which are dependencies. So genes which they actually, when they lose that function, they actually kill the cancer cells in a selective manner. And so you can also use sort of a Pac-Man analogy to think about this. So Pac-Man in this case is Cas9. You can imagine knocking out one gene and the cells grow a lot. So that's like their growth, that have a growth advantage from knocking out that gene. You knock out a second gene and they grow kind of average, kind of as is expected. But then you knock out the third gene and that leads to cell death in a specific population of cancer cells. And so that's what we're interested in studying. And this is not meant for you to read, but just for you to see how much this technology has really transformed therapeutic studies in pediatric cancers. This is focused on pediatric. If it was adult too, it would be a lot more than this. But this technology has led to lots of targets that have gone into clinical trials for new drugs and have led to, in some cases, treatment changes for children, which is really exciting. So how do we apply functional genomics in my lab? And sort of the second half of my talk is going to be focused on that. So I'm going to share with you stories that involve these two different principles here. So the different concepts of how we can apply CRISPR screening in functional genomics. So one is investigation of enriched dependency genes, what I just described to you from CRISPR screening in Ewing sarcoma and osteo. Today, I'm going to share with you a story in Ewing sarcoma with a goal of developing novel therapeutics. So we find a new gene that's a dependency. It doesn't necessarily have a drug, but then we can pair up with chemical biologists to develop new drugs. And the second is, can we use CRISPR screens to optimize therapy for these bone sarcomas? In this case, it's DNA damage repair therapy, which is something my lab is very interested in. So to give you this first vignette, I'm going to tell you about a gene called DDX19A, which is a selective dependency in Ewing sarcoma that we believe represses this EWS fly-driven gene transcription that I've already told you is very abnormal in Ewing sarcoma cells. And this project has been led by a very talented postdoc in my lab, Dr. Saikat Chakraborty, who's shown here. And you may think this face is familiar on the right-hand side, Dr. Tommaso Caputo in CBT, who I believe has already given you a lecture in this course. So he is our chemical biology partner for this project. So I'm first going to tell you a little bit about DDX19A. DDX19A is an RNA helicase that is a member of a large conserved group of dead box helicase proteins that basically are RNA helicases. They bind RNA. And I'm showing you here this phylogenetic tree of all of these dead box helicases, so you can kind of see how many there are. DDX19A is here, but there are sort of different levels of homology between them, but they're overall very well conserved, and many of them play redundant functions in yeast cells and in mammalian cells. This protein is not very well studied in cancer, actually, but we know in non-cancer studies that it binds and hydrolyzes ATP. It remodels RNPs, which are ribonucleoproteins, and it actually plays a key role in shuttling mRNAs out of the nucleus. So after mRNAs are synthesized, the DDX19A helps to shuttle them out of the nucleus for translation. It interacts in yeast cells with RNA polymerase II, so it has direct relationships to transcriptional biology. It certainly has not been studied in Ewing, and as I just said, it's really not been studied well for its functions in cancer. So this family has been targeted before. So I'm just showing you two examples of drugs that have been developed that are targeting dead box helicases. I am not a chemical biologist, as I said on my first slide, so I'm not going to get into the chemistry of this, but this is a very challenging group of proteins to drug, and we're really lucky that Tommaso is really a specialist in this area, Dr. Caputo. So we are working closely together with him to drug DDX19A. But these DDX2 family proteins that I'm showing you here have actually gone into clinical trials. So there is really hope and like a path for these types of proteins to be targeted in cancer. So I'm showing you here what is an example of output from a genome scale CRISPR dependency screen from the DepMap. So here I'm showing you a bar and whisker plot of each individual dot that would be, I haven't shown the dots here, but would be shown on the plot here, are individual cell lines that have been screened. So in this case, we have 27 Ewing sarcoma cell lines and then 1,100 or so other cell lines. And this is what's called a Chronos gene effect score, which is seen on the x-axis here, which basically assesses the relative dependency of an individual gene. So basically, how much is this gene, in this case DDX19A, killing these individual cancer cell lines. And the more negative the score, the more it kills those cell lines. So you can see that the box is shifted towards the left in the Ewing sarcoma cells, and that, there's some outliers in this other group, but for the most part, these 1,100 cancer cell lines cluster more to the right of the Ewing sarcoma, and this is very significant. So we noticed this in the data, and we were very interested in this, partially because of Dr. Caputo's expertise in this space, and so we decided to investigate this a little bit more. And the first thing we do when we have a dependency from CRISPR screening is we need to validate it, right? This is a big screen. It's a screen is by nature a large scale endeavor that doesn't always pan out. There are examples of CRISPR targets that are, off target effects for whatever reason. And then when you go to test them in the lab, you find that they're not real. So the first thing we had to do is validate this at low throughput. The way we did this is by using CRISPR guides to individually knock out DDX19A. So I'm showing you two different CRISPR guides here, number 1 and number 2, in each of these 4 cell lines. And this is AAVS1 is what's called a gene desert control. So it targets a region in the genome that doesn't have any functional gene expression. And so it shouldn't harm the cells by knocking out that area. And so you can see the DDX19A protein levels here, that they're reduced after you knock out 19A. They're reduced over here to various amounts. And what we saw was really striking. In the green box, I'm showing you 2 Ewing sarcoma cell lines. And you can see those cells are dead as doornails. Like really, they don't grow after 2, 4, 6, out to 8 days. Their growth is very stagnant in both of these cancer cell lines. But I'm also showing you here a neuroblastoma cell line and an osteosarcoma cell line. And so you can see in other cancers, they don't really care. So this does seem to work in Ewing sarcoma, and it does seem to be somewhat selective to Ewing sarcoma, at least as compared to these other tumor types. Next, we wanted to sort of validate like what the effects were. We were seeing these cells die, but how are they dying? And so on the left-hand side, I'm showing you the output of a flow cytometry experiment that's looking at cell cycle by staining the cells for something called propidium iodide. And so propidium iodide stains DNA. And so these cells are fixed in a way that allows their DNA to like basically the membrane is dissolved in the cells. So you're able to stain the PI directly. And what you're basically quantifying is the amount of DNA in these cells. And so these are cells that are mostly alive, right? And so what these peaks are showing you is that there's a large amount of DNA that's present at this peak. And then you sort of have this shelf here, and then you have a second peak here that's smaller, that's roughly double here compared to what it is at 100K to 200K, right? So it's roughly double. So what you can see here in this control is that most of these cells are sort of in G1 phase, which is what it would be expected. S phase comes next, and so that's the sort of shelf here you see. And then in G2 phase, they've doubled their DNA content, and so that's how you can tell that that's G2 going into mitosis. And so that's what you would expect to see in dividing cells. And so we have these controls over 24, 48, and 72 hours after DDX19A depletion. And that's represented by here. This DTAG is basically a tool that we use to deplete DDX19A that I'm not going to get more into in this talk. But you can see that gradually this G2M peak explodes after you have a loss of DDX19A. And so what we would say is that there's a G2M arrest in these cells, that basically they're not able to move through into mitosis, and they're arresting this G2 phase. Along with that, we can use flow cytometry to identify, both using propidium iodide, similar to what we use in this cell cycle assay, along with annexin. which binds phosphatidylserine on the outer plasma membrane of these cells as they start to die. So the phosphatidylserines or PS sort of gets externalized, and that's how you detect early apoptosis. And you can see in these bar graphs that after treatment with the DTAG, again, that you have a large uptick in these apoptotic and dead cells. So we think that there is apoptosis happening as well. The next thing we sort of wanted to see is what genes were affected by DDX19A loss. You know, we knew it was involved in transcription. And so what we were sort of surprised to see actually is that quite early on, before the cells are even dying, we had a massive upregulation in gene expression in these cells. And so you can see these are the upregulated cells. This is an RNA-seq output plot here. And you can see up and then down regulator genes is much fewer. And these genes in the middle that are gray are not significant. And so we were sort of interested in this because it was pretty non-specific, but the pathways that these genes are involved in, so we can take all of these upregulated genes and put them into a pathway analysis. And you can see that these are really largely involved in RNA processing and protein translation. So really, like it seems like the protein transcription, the gene transcription rather of mRNA, and then translation is getting completely messed up in these cells after DDX19A loss. So I'm not going to go any further with the mechanisms of this, except to say that this really piqued our interest about because of the nature of Ewing sarcoma. So basically what I told you before when I was introducing Ewing sarcoma is that you get aberrant activation of genes, right? So you get activated enhancers in Ewing sarcoma cells that shouldn't be there and would be usually closed and turned off. And then you actually also get repression of some genes. And these GGAAs really, I didn't go too much into this, but these are actually the sites on DNA where EWS fly binds. And so typically we know that GGAA repeats or microsatellites lead to activated gene expression when the EWS fly transcription factor binds there, that it leads to activated gene transcription. Whereas if it's a GGAA singlet, so just one GGAA, it generally leads to suppression. So our hypothesis after seeing this RNA-seq result with this massive upregulation of transcription that we're now really ironing out the details of is that we think that DDX19A is really disturbing the normal balance that is seen in the Ewing sarcoma cells. So it's actually acting in its normal state. DDX19A is acting to repress some gene expression, either cooperatively with eFly or through some other way. And so when you disrupt DDX19A, you get this really aberrant upregulation in transcription, and then the cells can't handle it and they die. And so that is sort of summarized here in this working model where you have EWS fly that's sort of naturally repressed by DDX19A. There's sort of a yellow light, right? So there's a good amount of transcription happening, not too much, not too little, and that allows to happy, normal Ewing sarcoma growth. But when you delete DDX19A, whichever way, with hopefully with a drug in the future, you get this upregulation and there's tons and tons of transcription because these breaks have been taken off. And that leads to what we're calling transcriptional overload and cell death. And so just to show you a snippet, we do have some drugs that we've been developing with Tommaso that we've started to test in cells. And these are just two examples. And what I just want you to appreciate here is that there is a dose-dependent killing of Ewing sarcoma cancer cells. And so we're really excited about that. And these are far from being real compounds that we can use to test more broadly. But they're a start. And so now there's a lot of chemical biology going on that's very exciting to refine some of these molecules and optimize them for further studies. One of the things that they're not optimized for are in vivo studies, but because we have other tools available to us in the lab, we used another tool, this DTAG tool that I mentioned previously, to degrade Ewing sarcoma cells degrade DDX19A in Ewing sarcoma cells. And we put this into, we implanted these cells into mice. So you can see the two pictures of these mice below, and you can see this big tumor on the side of the mouse flank. And we treated them with this degrading small molecule, so it degrades DDX19A, and we have cured mice of Ewing sarcoma using this, which is really exciting and not something we get to say too often. And I can say that because I'm only showing you this out to 60 days, this Kaplan-Meier curve, that all these mice are surviving happy with no tumors. As you can see, one mouse down here with no tumor, but now it's really out to almost 4 months and they have not regrown tumors and they're off treatment and they're just living and living their happy mouse lives. So that's exciting to us. So we're really working to pursue this target. Okay, I'm going to switch gears here a little bit and talk about a second vignette that I think illustrates a very different way to use functional genomics in biology. And this is looking at how to optimize PARP inhibitor sensitivity in osteosarcoma. So the other bone tumor that I study using CRISPR screening. And this project has been led by Janelia Moresby, who's another excellent postdoc in my lab. And so, back to the DEP map and the CRISPR screening, one thing I didn't tell you was that in osteosarcoma, if you look at all the enriched dependencies, if you kind of get a list of all the enriched dependencies in osteosarcoma versus everything else, all those other 1,400 cancer cell lines in the DEP map, and you look at these by pathways, one of the things that scores very highly is DNA damage. And this may make sense to you, right? Because I told you the genomes are very scrambled of these osteosarcoma cells. They need to somehow replicate themselves through all this chaos. So we think that DNA damage repair is essential in these cells. And so my lab is really interested in ways to target that and target it better in osteosarcoma patients. And so, when we think about DNA damage repair, I'm just showing you a very brief overview slide here just to illustrate there's a lot of different mechanisms by which cells repair their damaged DNA. And these are things that your cells are doing all the time. Normal cells are doing all the time, right? So and these are sort of the things or the agents that can cause these different types of DNA damage at the above. So like, ionizing radiation, chemotherapeutics is a big one, which obviously you wouldn't be getting unless you have cancer, but UV light is a big one. We're all exposed to UV light when we're out in the sun. And so our cells have ways to repair DNA damage. And the one that I'm going to focus on really here today is double-stranded breaks. Which is traditionally taken care of by a mechanism called homologous recombination, or HR. So next, to sort of give you a background of what a PARP inhibitor is, I think all of you probably have heard of PARP inhibitors because they're pretty prevalent in cancer therapeutics. But so if you have some DNA damage shown here in this cartoon, that leads to a single-stranded break, PARP1 gets recruited to that single-stranded break to repair it. And so fine, that's all and well and good. But if you have a PARP inhibitor in the mix, it cannot repair it. And so you get what's called PARP trapping, which basically means PARP is stuck to this site of a single-stranded break. It cannot be released. And this leads to a double-stranded break. And then there are two things that can happen. Either homologous recombination comes in and repairs those cells, or in the case of some type of deficiency in homologous recombination, it leads to cell death. And this deficiency is precisely what we're trying to capitalize on with PARP inhibitor treatments. And so this is what we call a synthetic lethality. So in a case where a cancer cell is not able to perform homologous recombination, or HR, it's a prime place to use a PARP inhibitor because that is it really optimizes PARP inhibitors use case. Most commonly in adult diseases, the HR deficiency comes from the loss of two genes, BRCA1 and BRCA2, which we don't really see in pediatrics, but I'm just giving you those genes here for just to help you remember, because these drugs are used a lot in breast cancer and ovarian cancer patients where those are found. Okay, so will osteosarcomas respond to PARP inhibitors? So osteos, as I said before, pediatric in general, do not have loss of BRCA1 or BRCA2. However, There are many computationalists who have derived signatures of BRCA likeness, which basically means do abnormalities in BRCA pathways exist that would lead to the same outcome of homologous recombination deficiencies. And these exist in significant subsets of osteosarcoma tumors because of their copy number changes and structural variations, as well as mutations. So this means, and a lot of people have thought about this before me, that osteosarcoma should respond to PARP inhibitors. Why wouldn't they? But the data has been really mixed. And there are a few patients who have been treated with PARP inhibitors, but not really systematically in osteosarcoma. So I'm showing you a couple of examples of preclinical data here, but just trust me that there have been some successes and some failures with these agents, and we don't really understand why. And then just to point out, this is just a general principle in cancer therapeutics, that any single agent, in this case PARP inhibitors, will eventually lead to resistance. So it's probably better to develop combinations anyway. And so we wanted to answer the question, can we enhance sensitivity to PARP inhibitors in osteosarcoma in a way that could be tested in clinical trials? And reminder, this is again, we're talking about CRISPR screening as a way to answer that question. So I've already shown you this schematic of a genome scale CRISPR screen. I'm now going to switch that out for a slightly different schematic, but just to show you it's the same principle of infecting cancer cells. In this case, two different osteosarcoma cell lines, Cal72 and SAOS2, with this genome scale CRISPR library. You take your infected cells and you let them grow for 14 days in this case, but in this case, you divide them into two arms. One arm is treated with a PARP inhibitor and the other is not. And the goal is to see where that enriched dependency is, that dropout is even more in the presence of, in this case, olaparib or a different PARP inhibitor. So in other words, what genes, when they are knocked out, sensitize you to the PARP inhibitor and make the cells die more? Because perhaps if we find out those things, we can put together a drug combination that would target that gene and then lead to a more successful treatment. So just as we're defining drug synergy here, this is a really basic schematic, but just to remind you that when we talk about synergy, we're talking about more than drug A plus B. So this dotted line would represent the effect of A plus B. And in this case, a synergistic case, you have an excess over additivity. So there's more than an additive effect on cancer cell death, there's a synergistic effect on cancer cell death. And this principle is used a lot in studying cancer therapy combinations because you want to both be able to reduce the drug doses of drug A and drug B and still achieve a magnitude of effect that's successful. And, reducing doses gives you less toxicity and all of that good stuff. So that's what we're trying to achieve here. So this is the output of the screens that we did in these two cancer cell lines. One, SAOS2, is represented on the y-axis here. One, Cal72, is represented on the x-axis. And this is a graph showing what's called the STARS dependency score. You can liken it to the Kronos score that I already explained to you, but it's computationally a little bit different. And this is looking at basically what is in this box enhancing sensitivity to that PARP inhibitor, and in this box up here, what are the makes it more resistant. And one of the things that we were really excited about, just looking at this data right off the bat, is one of the only things that makes cells resistant to PARP inhibitors is PARP1, which is a nice sanity check, because if you delete PARP1, you shouldn't have an effect of a PARP inhibitor. So it was nice that we saw that. The screen worked. And then we had a bunch of targets here, but one that caught our eye was ATM. And so I'm going to talk to you a little bit about ATM. So, and why we would want to go after this. So ATM is an enzyme that's involved in how cells repair their double-stranded breaks. So I told you the PARP inhibitors lead to double-stranded breaks, and then that HR needs to come in and repair those double-stranded breaks, otherwise the cells will die. So it might make sense, right, that if we're inhibiting something that that repairs double-stranded breaks, you would get enhanced cell death, and that's just shown in this schematic here. The second reason why we wanted to look at this is because there are selective ATM inhibitors that are in clinical trials. So this was a readily translatable combination to us that we could potentially bring to the clinic for patients. And just, in case you're wondering, there is a lot of safety data for PARP inhibitors in pediatric patients. So we've tested PARP inhibitors a lot in pediatric patients, and that's really a nice hurdle to have cleared when thinking about putting it into a new pediatric population, because otherwise it will take a really long time to put together a drug combination for like a new drug combination study. Would be a very hard thing to pull off. But since PARP inhibitors have great safety data, the fact that these ATM inhibitors have only been tested in adults really doesn't matter as much. We could potentially put this into kids. And so that made us excited. So again, we're looking for this synergistic effect, and we went on to test this. Again, you have to validate the findings of your screen, right? So we tested an ATM inhibitor plus a PARP inhibitor in a battery of cancer cell lines, and I'm just showing you 2 osteosarcoma cell lines here. And what these schematics are showing you are escalating doses of PARP inhibitor on the y-axis, escalating doses of ATM on the x-axis, and that's the same is true here. And the more red these boxes are, the more bliss synergy, which is one of the measures we use to measure synergy, there is. And you can see that there's a pretty broad swath of red here that's pretty synergistic. Same here in this other cancer cell line. And another way to look at this is sort of similar bar graphs to this schematic I showed you up here, but this is representative of one dose combination that is the max synergistic. So whatever the darkest red boxes are here, and maybe here. But so you can see the effect of one single drug at that dose. You can see the effect of one of the other drug at that dose. And then you can see, and in this case, it's actually pretty cool because they're both, actually for both of these cell lines, that's true. There's almost like a negative effect, right? So these drugs are really not having any effect, but when you put them together, they have a really nice synergistic effect. So we were really excited about this. Another thing when talking about drug resistance that excited us and really excited me was that we had cancer cell lines, we had osteosarcoma cell lines that were really resistant to a PARP inhibitor. And we put an ATM inhibitor just on them just to see if they would sensitize. And so what I'm showing you here are outputs from viability experiments that are sort of complicated to understand, but I want you to focus on the IC50 or the median cell killing doses here. So in this cell line with talazoparib, which is a PARP inhibitor, you have an IC50 of 0.2 micromolar, which is not terrible, but it's still decently high for this. In IC50, like could not even be determined in the ATM inhibitor because it wasn't efficacious at all. But when you put the two of them together, it's much lower. It's 0.01 micromolar. So you're getting nice synergy or enhanced efficacy there. And the same is true as here. It's probably like it's even more of a reduction. So we were really excited about this because when we do clinical testing in early clinical trials, we start in refractory and resistant patients. So patients who have seen a lot of drugs, who are resistant to a lot of things. So this may help us to mimic that setting, and we might be able to achieve some success in those patients. Here I'm just showing you the overarching synergy by a mean adjusted synergy score, and you can see that the majority of the osteocell lines in red pass that mean adjusted synergy that we would expect for to call synergy, whereas other cell lines, in this case 2 neuroblastoma lines and a fibroblast line, do not. So that is relatively specific to osteosarcomas. We have 2 markers here of apoptosis, caspase-3 and annexin, which I mentioned earlier. And you can see that the staining of those in these cells, this is done by live cell imaging using a live cell imaging dye, go way, way up for both of these markers of apoptosis in the combination compared to either single agent down here. So that was really suggestive to us that the cells are dying. And then, as you would expect, by adding 2 DNA damaging agents together, you see more DNA damage. So this is immunofluorescent imaging, which you probably have all looked at before in some paper or other, using 53BP1 and gamma H2AX, which are 2 markers of DNA damage. And you can see there's some foci, because you always have DNA damage being repaired in cells. But then when you add an ATM inhibitor, maybe not that much change, maybe a little bit of change. PARP inhibitor, you do see some change, but then you see a ton of change in the combination, and that's quantified here in these bar graphs. So an amalgamation of cell death by apoptosis and increased DNA damage. And so the last data shot slide I'll show you here is a combination study that we did in mice, because obviously that is a precursor to clinical trials. And this is just a way the trial was done. We used these different doses. AZD 1390 is the leading ATM inhibitor that's being used in clinical trials. AZD 5305 is a PARP1 specific inhibitor that's at the forefront of the line of PARP inhibitors, the more selective PARP inhibitors that are being developed. And so we treated mice, we randomized mice that had tumors to receive either like vehicles for both of these drugs, one drug, the other drug, or both in combination. And then we monitored for toxicity, obviously, we monitored for tumor growth, and we collected tumors, and now we're doing lots of follow-up studies on those tumor samples. But you can see that the combination modestly extended survival in the combination. So it's statistically significant. I call it modest because it is. It's not curing mice like I showed you in the first vignette, but we think it's a starting place. And now we're working on doing further refinements of the doses of these two agents, and looking at biomarkers to determine which osteosarcoma tumors will really respond best. Okay, so just returning you back to sort of an overview. I hope I've sort of illustrated these different parts of what my lab does. So we use a lot of functional genomic screening, either in our own lab or as part of the PEDDEP or DepMap project, to try to identify through CRISPR and other omics what are these selective dependencies. So really getting our ideas from these selective dependencies. And we then do perform investigation of these dependencies using different models in the lab. Modeling perturbations in mice is an important one that I've highlighted for you here. And obviously the overarching goal for me as a physician scientist is clinical translation. So can we take this into patients and help them cure more patients? So with that, I just like to thank the people who did all the work that I told you about today. So these are all the people in my lab. I've mentioned Saikat and Janelia by name already. John Harper is a very skilled in vivo technician who does all of the mouse work in my laboratory. And so he did all those studies that I showed you. Our collaborators, most importantly, Tommaso and Paul Gilliher, whose lab really helped us with the drug synergy project that I just showed you. Sona is an undergrad student, just to inspire all of you undergrads out there, that we have a lot of undergrads that come to St. Jude campus and do research over the summer. And she actually spearheaded the PARP ATM project that led to Janelia's more finished work that we're about to publish now. And so kudos to her and all these other people here. And of course, we depend on lots of funding to do the work we do. And I've listed all of our funders here. I'd love to answer questions if there are any.
All right, thank you. So there was a question that appeared earlier on. It's has to do with where you had mentioned that the osteosarcoma has difficulty being treated by radiotherapy. So the student asked, could chelation therapy help with the calcification that's presented? Has it been tried for potential treatment?
Has what been tried? Sorry, I didn't hear you.
Chelation therapy, I'm assuming metal chelation for the calcium calcification.
I think so, but I have to say I'm not very knowledgeable in that space. I know there are people who are trying to use, for example, focused ultrasound therapy to try to break up some of the calcification. So there are different types of ultrasound waves that have been developed for some of these more calcified tumors. But you can imagine that maybe chelation would be helpful, but I'm not really familiar with the studies that have been done in
Okay.
that case. I'm sorry.
Oh, all right. Are there any other questions from students? Let's see. Okay, there's another question. It has to do with the animal model. So it said if there was no good animal model for Ewing sarcoma, how was the xenograft model created?
Yeah, that's a great question. So when I was referring to there are no good animal models, what I was actually referring to was there are no models that are possible in immunocompetent mice. So
Mm.
mice with an intact immune system. Because in order to do that, you'd have to actually manipulate, genetically engineer somehow. I mean, there are some ways around this, but for simplicity's sake, the way you would like to develop a mouse model that has an intact immune system, which is really important, obviously, for the cancer microenvironment, studying immunotherapies, et cetera, um, would be to genetically manipulate the actual progenitor cells in the mouse. And so, since we don't know what that progenitor is, we have had a hard time. There are whole papers talking about failures of many, many very important labs in the Ewing sarcoma field who have tried to do this. So many very smart people have tried to put EWS fly transcription factors into different progenitor cells in mice, and they can't get it to form tumors. So
Oh,
it's
okay.
a really interesting problem that we haven't been able to overcome in that space. The xenograft model that I showed to answer that question,
Hmm.
though, was done with an immunocompromised mouse, so a mouse that doesn't have it, with its bred to not have an immune system, and so it doesn't reject human cancer cells. And so you implant into that mouse, in this case, the flank, but you could also do an orthotopic implant into the bone of the mouse to mimic better, even better, a bone tumor. Um, uh, a series of human cells that then can grow into tumors, human tumor cells that the mouse won't reject. I hope that answers your question.
Okay, that's great. Are there any other student questions?
Um, I have a quick question. It's
Sure.
more of like a hypothetical.
Yeah.
Um, could you, like in theory, keep doing CRISPR screenings with your combination therapy? Like first the PARP inhibitor, but then just keep adding drugs to that mix to see if you see more synergistic effects?
Yeah, totally. So you're talking about you have a PARP and an ATM inhibitor on board, doing another CRISPR screen to determine like the third agent that you might use or the fourth.
Exactly.
Yeah, so there you could. I think, when one of the things we try to balance in cancer therapeutics is, we know we're always going to have to use combinations of drugs. There's always a trade-off between multiple drugs, getting drug synergy, minimizing toxicity, but then when you add more and more drugs to the mix, you get even more toxicity. And so, the for better or worse, targeted combinations, so single, like two drug combinations, are what have been trialed for the most part. There are definitely applications of this, and this is something we've started to think about a little bit, because particularly, you're probably not going to test these agents, especially up front in osteosarcoma patients, without chemotherapy as a backbone, right? Because it wouldn't be ethical to not give them the chemotherapy that we know works. And so, and you would imagine that with two DNA damage repair inhibitors, that if you give them a DNA damaging chemotherapy agent, then it exacerbates their function, their effect rather. And so I think, we've thought about, like, do we add in cisplatin to this? Do we add in doxorubicin, which is another drug that's used to treat osteosarcoma patients? And that might be a more realistic combination rather than doing a CRISPR screen to determine another targeted agent.
Thank you.
But in
And
principle, you're right. Yeah.
yeah, any additional questions from students? All right. So if there are no further questions, I wanted to give a round of applause to Lily for such a great talk. Thank you so much. Very insightful.
Thank you so much for having me. And I, do they have my contact info? If you could send my email around if
Sure,
there are
I'll do that. Yeah, that'd be
questions,
great. Yeah,
or if anybody wants to talk more about cancer biology, I'm happy to be a sounding board
yeah, that's
resource.
amazing. All right, thank you so much, and have a great weekend. All right, take care,
Okay. You
everyone.
guys
All right,
too. Take
all right.
care. Bye.
Bye-bye.